we can move to the next and last speaker. So Laura Heraty. So she's a PhD student at Cardiff University in Cardiff, and we'll talk about FAN1 prevents CRISPR-Cas9 Nickase-induced contraction of CTG repeats, and also she has a poster C2. Thanks.
Okay, hi. I want to say thank you to the organisers for allowing me to present some of my work. So I'm just going to jump straight in. So the work, what we are interested in our lab is CAG or CTG expansion disorders. So currently, expansions at 16 different genomic loci cause a range of neurodegenerative and neuromuscular diseases, and all of these are currently without disease-modifying treatment, and many of them exhibit similar traits, whereby the longer your repeat length, the lower your age of onset, and also the more severe the disease phenotype. So because of this, contracting CAG or CTG repeats to non-pathogenic levels could be a potential therapy, and that is the sort of work we're doing in our lab. So we have a system to contract the repeats using a modified CRISPR-Cas9. So this CRISPR-Cas9 is a Cas9 nickase, whereby one of the nuclease domains has been silenced, and this allows us to nick and create single-strand breaks across either the CAG or CTG repeats. And we've shown this in a few cell lines and essentially shown that using the Cas9 nickase, we see contractions to non-pathogenic lengths. However, the mechanism by which this contraction occurs is not well defined, and that's a major focus of our lab, is to try and understand how contractions occur, which is important not only in identifying the mechanism, but it could be useful to screen for patients that would benefit from the therapies. As we know, many patients have different DNA repair variants, which could influence how effective the therapy could be. So a way that we initially screen for modifiers of Nickase-induced contractions is using a model HEC293 system. So in our HEC293 lines, we've stably integrated a GFP reporter, which contains an ectopic CAG repeat. The repeat resides in an intron that separates two GFP exons, and essentially this reporter works whereby repeat length changes to be measured using GFP intensity. So if you have expansion events, this interferes with functional mRNA splicing, reducing your GFP intensity. So then when we induce contractions with the nickase, this increases functional mRNA splicing, and we can see a higher GFP intensity. So we do a few then downstream analyses in the lab. So the one I'll focus on today for sort of time's sake is a small pore PCR, which is quite a classic method, essentially where we isolate DNA and dilute to just a few genomic equivalents and do a Southern blot, and this allows us to look at a spectrum of changes across the repeat tract. And these are just demonstrative examples. So is that pointing? Yeah, on the left-hand side panel is cell lines with 101 CAG repeats, and you can see each band represents a different allele. And then the right-hand panel, this is after treatment of the nickase, and you can see multiple contracted alleles to non-pathogenic repeat lengths. So the sort of focus of my project is FAN1, and we had an excellent intro to FAN1 earlier, so I won't really cover this, but just to say that we know it's a modifier of repeat expansions, and current literature suggests this acts to protect against expansions using a nuclease mechanism and also for its interactions with MLH1. So to explore whether FAN1 impacted Nickase-induced contractions, I generated some FAN1 knockout lines, and looked at the GFP, and the GFP indicated an increased GFP expression, and then I did subsequent small pore PCRs. So you can just see some demonstrative plots at day 0. You can see there's a comparative starting CAG repeat length, which we've sequenced, which is around 140 or so repeats. And then this is a panel at day 21 after treatment with the Cas9. So you can see both plots show contractions to non-pathogenic levels, but I think you can see quite strikingly that the FAN1 knockout really increased the contractions rates. And we quantified this, and you can see in the FAN1 knockout line a significant increase in the contraction efficiency over 21 days. And as recent literature has shown that FAN1 acts in somatic expansion through its interaction with MLH1, sort of downstream work we thought to look into would be to see if MLH1 plays also some sort of role in contractions. So part of the work I've done so far to do this is to create FAN1, MLH1 double knockout lines. And these are just demonstrative plots here again. And you can see at day 0, there's a starting CRG repeat length that is comparable. And you can see further contractions in MLH1 to the point where we almost lose the starting CRG repeat length. And we quantified this, so you can see wild-type FAN1, FAN1 knockout, and then the double knockout on the right further increases contractions to the point where roughly 85% of the cells have undergone a contraction event. And that's just to sort of summarize what I've taught you so far, is that in our system, a loss of FAN1 actually is beneficial to Cas9 Nickase-induced contraction rates, and we see an increase in contractions compared with the FAN1 wild-type lines in this ectopic CRG repeat. And this was further increased in a FAN1 MLH1 double knockout line. So obviously this is a reporter system, so ongoing work at the moment is needed to establish whether this is the case in non-replicative HD iPSC-derived neurons, and this is ongoing. And if you'd like to look at my poster, it's C2, and just if you have any other questions. And thank you to my supervisors, the Dion Lab, the Cardiff HD group, and again to the organisers for allowing me to present some data today.
Many thanks, Laura. So, any question from the audience? Are you still alive?
Yeah. In the meantime, I have a question.
Okay, thanks.
I was very intrigued by the data. MLH1, have you considered looking at loss of function of that?
Yes, so that was the plan. The CRISPRs were done at the same time. The MLH1 CRISPR took a bit longer to do, so those experiments are ongoing now, and I hope to have the data in the next month or two. So yeah, we'll be interested to see what that shows.
Very interesting indeed.
Okay. If there are no more questions, I would like to thank again Laura.